The penultimate chapter in Unit 4, Topic 1 is about inheritance. So after this, all we have to cover is biotechnology, which we've been teasing throughout, and then we are officially moving on to the last topic of Year 12 content. Inheritance is one that you've all covered previously. It's a big part of the Year 10 National Curriculum Syllabus, and so you're all likely aware of it and have worked with a lot of the stuff that we're going to cover in this. So for that reason, this is going to move through pretty quickly. There are only two objectives for inheritance, and they are both around predicting. So the first one is predicting frequencies of genotypes and phenotypes using data from probability models by taking into consideration patterns of inheritance. Effectively, what it's asking is, can you construct and use a Punnett square in order to determine the genotypes and phenotypes, or the likelihood of those genotypes and phenotypes from Mendelian genetics with autosomal dominant, sex-linked traits, and multiple traits, multiple alleles as well. We will also look at instances of codominance and incomplete dominance as well. Defining polygenic inheritance and predicting frequencies of polygenic genotypes and phenotypes is going to be the new thing. That's going to be the extension on what you already know. Some key definitions here that hopefully you remember. Gene, we've talked about. Allele, we've talked about. Genotype, phenotype, we've talked about. And then dominant, recessive, homozygous, heterozygous will come up a lot in this. But again, should mostly be known knowledge to you because all of this stuff has been previously covered. So inheritance, also known as heredity, although they are slightly different. Heredity is the study of how, and inheritance is the study of how characteristics and traits are passed from one generation to another. Not to be confused with hereditary, which describes something that can be passed on. This entire field was basically fathered in by Gregor Mendel, who was an Austrian monk, and back in those days monks got bored and they had a lot of spare time, and they did a lot of experiments. So Gregor Mendel particularly did experiments on pea plants, and one of the most impressive things about his discoveries is at the time they had no idea about DNA, they had no idea about genes, so they really didn't understand any of the mechanisms behind how this process worked. He looked at 7 particular traits in pea plants: seed color, seed shape, seed coat color, pod shape, flower position, and stem length. So whether they were short or tall. So he looked at those 7 traits, and through hand pollinating, which is an incredible amount of work, 29,000 plants, so that he could control the way that they bred specifically. So through those experiments, he came up with effectively what we know of the 3 laws of inheritance now. And the way that he tracked the inheritance of these particular traits, or one way that we can see how he tracked it, is through the use of Punnett squares, which you can see here. So if we use the example of seed color, you can see that a yellow individual has 2 Y, uppercase Y alleles. Okay, its genotype is big Y, big Y. Its phenotype is that it's yellow. So remember, the genotype is the actual alleles, the genes that the individual has, and the phenotype is how it's outwardly expressed. So yellow, big Y, is dominant, and that's represented by the capital letter, whereas green is recessive. Okay, so we had little y, that's the recessive alleles, and its phenotype is green. Now, when these 2 individuals were hand pollinated by Gregor Mendel, he found that all of the offspring were yellow. And through our Punnett square here, you can see that they all have 1 big Y, 1 little y inherited from each of their parents. And these guys having 2 different alleles are what we know as heterozygous. Hetero meaning different, zygous meaning zygote, because we're looking at what different alleles the zygote would have had. So the parents were homozygous for the dominant and recessive trait respectively, and the offspring were heterozygous. These are the three laws of inheritance. You don't need to necessarily remember these, but just to put it all into context. And I've got the sort of summary there in the red. The first law, the law of segregation, we have basically already touched on in the last couple of units. So each individual has two alleles for each gene, and they will inherit one from each of their parents. The law of independent assortment, we already know. Each gene is inherited independently of any other gene. So they're not inherently linked in any other way. And law of dominance states that some traits will be dominant, some will be recessive, and those recessive traits will be masked by the dominant traits or alleles. So each individual gets two sets of each gene from their parents, and they could be two different alleles, and that every gene is inherited completely independently, and that dominant traits will overtake our recessive traits. When we look at our most basic form of inheritance known as Mendelian inheritance often, or single autosomal gene inheritance, it's characterized by having a single gene controlling the trait, and it's found on an autosomal non-sex chromosome. In most cases, we have a dominant and recessive allele, and we can use Punnett squares to track these super easily, which is what Mendel did. You can see we've got a tall plant and a short plant here. Tall being a dominant trait and short being a recessive trait. So when we breed those two homozygous individuals, so tall and short in terms of their alleles, we end up with a whole bunch of heterozygous tall plants. So they're tall short in terms of their genotypes, and their phenotype is tall. If we then self-fertilize that plant, then the result of that is 3 tall individuals, 1 short individual. The actual numbers that Mendel ended up with was approximately 2.84 to 1. Not exactly a 3 to 1, but you're never going to get exacts in this sort of thing because, as we learned through crossing over, through independent assortment, random fertilization, a lot of these processes are completely random. So he found that the big T allele, the tall allele, causes the plants to be tall, the little T allele causes the plants to be short, and big T is dominant. Okay, so if any plants have a single copy of it, they will be tall, regardless of whether they have a short allele or another tall allele. So therefore, little t, the short allele, is the recessive gene, recessive allele, recessive trait. Okay. Now, the important thing to note here is Mendel managed to get lucky, and we are currently talking about only single autosomal traits here, single autosomal dominance. So this is a trait that is controlled by only one gene, and that is very rarely the case, particularly with organisms like humans that are complicated. Just a refresher of some of the terminology. So parent generation, or the P generation, is from the very beginning. First filial generation, F1, is when we breed together those two parents. And then our F2, second filial generation, is when those within the first filial generation self-fertilize. So in the F1 generation, if we have two homozygous parents for the different traits, then the offspring in the F1 will always be 100% heterozygous, expressing the dominant trait. And then the F2 generation will always produce a mixture of tall and short with a 3-to-1 ratio. A homozygous individual will always exhibit the trait that they have the 2 alleles for, because they only have the one different type of allele, just 2 copies. Heterozygous individuals will always express the dominant trait. Okay, so dominant trait will be expressed basically in 2 out of the 3 scenarios. Whether we have a homozygous dominant individual or a heterozygous individual, then both of those will show the dominant trait. Whereas only the homozygous recessive individual will actually show the recessive trait, because it only is shown up when there's no dominant trait to mask it. Punnett squares, constructing Punnett squares, you should hopefully be familiar with. One couple of things to consider that are often misconceptions when looking at Punnett squares is that the dominant trait is not always the most common. I've seen plenty of people fall into the trap of assuming that dominant means most common. It's not the case. And then the other thing as well, as I mentioned earlier, most traits are polygenic, meaning that they are based on more than one gene. So assume we're dealing with something monogenic in our studies because we do tend to simplify it a little bit. If we have a look here, we're just looking at another example of our Punnett squares and our basic Mendelian inheritance. Okay, so have a practice of some of those Punnett squares and hopefully it all comes back to you. We're going to look now at a couple of instances of non-Mendelian inheritance. That's when we don't have straightforward single autosomal dominance, and incomplete dominance is a good example of that. So in incomplete dominance, if we have homozygous for the main trait, the dominant trait, then we will see that individual expressing the dominant trait. Homozygous for the recessive trait will show the recessive trait. But the heterozygotes, the ones that have a dominant and recessive, or one of each allele, will actually show incomplete or partial dominance, where the dominant trait doesn't entirely mask the recessive trait. So we end up with sort of a blend. Easiest example, if we've got red as dominant and white as recessive, then the blended or incomplete dominant heterozygote is going to be pink. Snapdragons are often used in these examples because they show a really clear example of it. And if we look at self-fertilization of the F1 generation, or in this case, the parents were heterozygotes, then you can see that we can produce one red, two pink, and one white. Now, this follows the same rules as our Mendelian inheritance in terms of we've got one homozygote for the dominant trait, two heterozygotes, and one homozygote for the recessive trait. But in this case, we have a third phenotype introduced as opposed to just the normal two. In this case, we will often represent it slightly differently. Instead of just using the capital letters on their own as we've seen before, we'll use an uppercase letter for the trait. So in this case, because we're looking at color, it's capital C, and then we use uppercase superscripts to show the alleles. So uppercase R for red and W for white. So incomplete dominance creates a third phenotype based on blending of our two traits. Then we've got codominance, which is when both traits, both alleles are equally dominant. There is no recessive trait. We have two different alleles, white and black, but both are equally dominant and therefore equally expressed. It creates a third phenotype, again, just like incomplete dominance. So those heterozygous individuals will have a new phenotype, but in this case, it's not a sort of blending of the two, but they're equally represented. So if you've got black and white chickens, then an example of codominance is speckled because they have both black and white feathers being shown in the phenotype. A very common example are roan cattle. Roan cattle exist when you have a homozygous white parent, homozygous red parent. So the individual, the calves, have both red and white hairs, resulting in each of them being expressed equally. Now, it doesn't mean uniform when we say that each of them are expressed equally, but they're just separately represented. Punnett squares is the number one way that we can represent our results from tracking inheritance. But one other way that you may see, and the only reason I say you may see this is because it's specifically mentioned in the syllabus, you could be given this frequency histogram and asked to basically analyze it in order to identify proportions or expected frequencies of different genotypes and phenotypes. We often see this represented in polygenic inheritance, Because it more accurately represents the spectrum that we get with polygenic inheritance. But you may see it used as an example for non or monogenic inheritance. All right, sex-linked inheritance, which most of you will probably more likely know color blindness as being a good example. So sex chromosomes are the only ones that have different genes on them. Okay, and because they have these different genes, we can have an example of hemizygous, which means there's only one copy of it in a diploid cell. So the X and the Y chromosome are very similar, apart from the Y chromosome dropping a leg. And when it drops that leg, it loses some genes. So traits inherited from genes on the sex chromosomes are sex-linked traits because they're inherited on the X and Y chromosomes. And the SRY transcription factor gene that we talked previously about is an example of this inherited on the Y. Whenever we have unequal phenotypic proportions between sexes, that trait is most likely sex-linked. So if you're looking at a bunch of data and it looks like one sex over the other more commonly has this trait, we're likely looking at sex-linked traits. Eye color in our friends Drosophila melanogaster, the fruit flies, is a good example of an X-linked trait. So if it's an X-linked trait, females are getting 2 X's, so they will get 2 copies of it. But males could only get 1. And in the case of eye color, we have red or white. Okay, so red being the dominant trait, white being the lower, recessive trait. So for a male can either be red or white. It only inherits one copy, one allele for that trait. So there's no heterozygousness. It's just hemizygous. A female could be homozygous dominant, heterozygous, or homozygous recessive. In both cases of homozygous dominant and heterozygous, the female will have red eyes. You can see a cross here between a red-eyed male and a white-eyed female. The result is we produce 2 red-eyed females and 2 white-eyed males. Some further examples there, but we're using big R, little r instead of big W, little w. When we're looking at X-linked dominant traits, as opposed to X-linked recessive, which are significantly much more common, X-linked dominant traits will show in homo and heterozygous females and all males. So what that means is that affected males will not be able to pass it on to their sons, okay? But they will pass it on to all of their daughters because they're donating an X to all of their daughters. X-linked recessive traits are much more common, and males often are more likely to get these traits because they only need one of that trait for it to be expressed, okay? So they only need one X, which they could inherit from their mother, which will carry the trait. In the example of color blindness, this is exactly what happened. So if we look here, in the example of color blindness for X-linked recessive, okay, if you have an unaffected father and a carrier mother, then that will result in a 50-50 shot for the son to be affected or not, and then a 50-50 shot for the daughter to be a carrier or unaffected. If the father is affected and the mother is unaffected, then none of the sons will be affected because they inherit the Y from their father. They're not inheriting the color blindness gene from their father. However, both daughters will be a carrier. It's exactly what happened in my case. My grandfather was colorblind, and so both of his daughters carried the color blindness allele. Now, neither of them were colorblind because they only inherited one of them. When my brother and I were born, there was a 50-50 shot. My brother ended up being unaffected and I ended up being colorblind. My auntie, who is a carrier, had 2 daughters. So what that means is that her daughters could be carriers or unaffected. Multiple allelic inheritance is not mentioned explicitly in the syllabus, but the definition of polygenic inheritance in the syllabus is confusing and potentially incorrect. So we include it anyway. And the most common example, and probably the only example you're likely to see, is DNA. Okay, sorry, not DNA, blood type. Now, this is not an example of polygenic inheritance in that it's only controlled by one gene, but it's multiple or polyallelic in that we don't just have the normal 2 alleles. We actually have 3 possible alleles that could result, as opposed to 2 for every example we've looked at so far. So our possible alleles are O, A, and B, and the result of that is 4 potential phenotypes. So the phenotypes are blood type A, blood type B, blood type AB, or blood type O. And the possible genotypes that we have from that are 6. So you can have homozygous A, or you could have heterozygous A, both of which will be A blood type. You can have homozygous B or heterozygous B, both of which will be the B blood type. Okay, and then you can have AB. So A and B here are both codominant, whereas O is the only recessive one. So in order to have O-type blood, you must have 2 of the recessive O-type allele. And this is a great example of dominant not being more common. We have 3 potential dominant phenotypes here, yet O-type blood is significantly more common, at least in Australia. So that's multiple alleles, and we will work through some examples of that in class. Okay. Dihybrid crosses, we looked at monohybrid crosses with our Punnett squares before, mono meaning 1, di meaning 2. Instead of looking at just a single trait, we can look at how 2 traits are inherited. So we can look at the genetic patterns of both of those traits. The result of this is basically just a 4 times as big Punnett square with some different phenotypic ratios. So we've got 9, 3, 3, 1. 9 for both dominant traits, 3 for dominant trait A and recessive trait B, 3 for recessive trait A and dominant trait B, and then 1 for recessive. It worked basically the same as a monohybrid cross Punnett square, but the difficult thing is working out what our alleles are that are being donated. So you need to look really carefully here at what are all our possible alleles. Now this one's pretty basic because both of these are homozygous for both traits, so they could only give 1 possible allele for each trait. But if you look here, this example, okay, then we've got big R, big Y, little y. So the possible combinations are big R, big Y, or little r, big Y, big R, little y, and little r, little y. So it makes it a little bit more complicated in that you've got more things to consider there. You can see an example of a dihybrid cross here, along with the second filial generation. All right, moving into that second dot point now. So everything we talked about previously are just different types of inheritance. Some of them 100% mentioned in the syllabus. Others you probably should know. But the main thing is, if you can only take one thing away from this, you need to be able to conduct a Punnett square, because that is how we're going to predict our genotypes and phenotypes. Our last dot point is polygenic inheritance. Poly meaning many, genic meaning genes. So these are traits that are controlled by more than one gene, which is the vast majority of human traits. Height in pea plants, for example, is controlled by only one gene with 2 alleles. And so you either end up with pea plants that are tall or short. There's no variation other than that. It makes it a discontinuous variable, tall or short. Whereas height in humans is a continuous variable, meaning that it shows a range of phenotypes. And not only that, it's also heavily influenced by epigenetic factors such as the environment. So if you look at the height of people and you represent it on a frequency histogram, as you can see here, you'll notice that there are people on all ends of the spectrum. Now, if we simplify it and say that every human has only 2 different alleles for height, tall or short, but there's 7 genes that are in charge of height, then that means for each of those 7 genes, you could have a tall or short. On the very bottom end of the spectrum, you would have somebody who has 7 short alleles, and the result of that would be that they would be very short. On the other end of the spectrum, you would have people who have 7 tall alleles, and they would be incredibly tall. Or you can have what's more likely somewhere in the middle where they've got 3 or 4 of each of those and represent, a more median height. So multiple genes and environment contribute to these, but it is polygenic, meaning it ends up being like a spectrum. And eye color is another great example of this. If you put blue eyes at one end of the spectrum and like sort of, dark brown, almost black eyes at the other end, then everyone fits somewhere along that spectrum with sort of green and hazel falling in the middle. Best and easiest example for you to access in terms of polygenic inheritance was the first one where it was noted in wheat kernels. So kernel color is controlled by 2 genes. Okay, polygenic or digenic here, just 2 genes. Each of those has 2 alleles, one for red color, which was dominant, and then one for white, which was recessive. Okay, so it was found that if you ended up with 4 big R's, 4 red dominant alleles, then that wheat would be dark red. Only 3 of these big R's, then we had a medium to dark red. 2 big R's and 2 little r's, then it was medium red. 1 big R with 3 little r's, then it was a sort of quite light red, and then no big R's, all little r's, resulted in a white individual. This is just color mixing like you will have done in kindergarten pretty much, okay? Depending on how much red and white you have, depends on the color of the outcome. And you can see that represented there in our frequency histogram, okay? So depending on what the outcome is and the mixing of those traits, then we can get a whole variety of different colors. Now the trick with this one was that they actually discovered later on that there was a 3rd gene responsible. So still polygenic, this time trigenic, okay? Each of those still comes with 2 alleles, one for red, one for white, okay? But in this example here, we're representing each allele with a letter, the capital being the dominant, which is the red pigment, and then the lowercase being the recessive, which is the no pigment. And you can see the huge spectrum we can get there. And this is effectively a trihybrid cross, whereas that last example we're looking at there is an example of a dihybrid cross. Now, it is probably unlikely that you'll be asked to construct something like this, even a dihybrid cross in an exam scenario, just due to the sheer amount of time it takes to do this. More than likely, you'll be given some information about this. You'll be given the Punnett square or a frequency histogram and asked to talk on that, okay? Or predict the outcome of, maybe some simpler mating pairs. That's it for inheritance. You will do a lot of practice on this in class, and that is the key. Practice, practice, practice. If you can do those Punnett squares, you are nailing it, okay? So we'll talk more in class. Hopefully this has been helpful, and good luck in your inheritance.